So, good morning, students of BT First Sem. Today, I am going to discuss with you about the microfilaments. Yesterday, I have taught you about the microfilaments. So, let's get started now. So, about the microfilaments, if I say that microfilaments, wait. So we will say about microfilaments. Well, microfilaments are primarily structural in function and are an important component of the cytoskeleton. Along with your microtubules and often your intermediate filaments, these microfilaments are also important in structural organization of the cell. Generally, these microfilaments ranges from 5 to 9 nanometers in diameter. It ranges from 5 to 9 nanometer in diameter and are designed to bear large amounts of tension. So they are the essential parts to make the cytoskeletal framework of cell. These microfilaments associate with myosin and they help to generate forces used in cellular contraction and basic cell movements. So, these microfilaments are involved in your cellular contraction. Okay, and your basic cell movements. Basic cell movements, right? Now, these filaments also enable a dividing cell to pinch off into two cells and are also involved in amoeboid movement of certain types of cell. So, during cytokinesis, the process of cytokinesis that occurs just at the end of your cell division process where the cell which is going to be dividing, they are pinching off into two different cells. Let's say the cell is dividing, a tension is being creating between the forces that pinch off into two cells. So this pinching off is also mediated by the movement of the actin filaments, movement of the microfilaments, right? And also the amoeboid movement in certain types of cell are generated your by these microfilaments. Now, generally, if I say about the microfilaments, they are a type of solid rods made up of a protein called actin. So there is a protein known as actin. Often we say microfilament as actin filament. So these are the proteins which make up the microfilament. Generally, when actin is produced by the cell, it appears in a globular form. So it is a globular protein. This actin is a globular protein, right? And I have discussed with you about the globular protein. What are globular proteins? Globular proteins are spherical proteins which are somewhat water soluble, right? And it is completely different from the fibrous protein. So normally these actin filaments appears in globular form, right? and they are called as G actin initially. When they are in loose form, they are called as G actin. Now, in microfilaments, however, which are often referred to as actin filaments, long polymerized chain of molecules are intertwined with one another in a helical form, creating a filamentous form of the protein called F actin. So, when these microfilaments they are polymerizing, okay, so these molecules called G actin are intertwining in a helical conformation, creating a filamentous type of protein called F actin. And all of the subunits that compose a microfilament are connected in such a way that have the same orientation. Thus, this microfilament will have a polarity. They have two end groups. You can recognize which one is which end. Okay, there will be, just like microtubules, there will be a plus end where assembly of the microfilaments are occurring, and a minus end where disassembly of the microfilament are occurring. Okay, but there is some difference between these microtubules and microfilaments. What is the difference? We will discuss now. So we have said that first individually we see G actin. When actin protein is first produced by the cell, it is called as G actin, and these actin filaments are then, these proteins are joined with one another. They form long polymerized chain of molecules of microfilaments, which are called, and now we will call them as F actin, filamentous actin. And they have a polarity. I have said microfilaments have a polarity because the molecules join in the same orientation. The actin molecules join in the same orientation. Not only the actin molecules, other proteins are also associated, okay, which are non-actin proteins, and they have a plus end where assembly of the microfilament occur, and a minus end where disassembly occur. But unlike your microtubules, generally we know microtubules typically extend out from the centrosome of a cell. Microfilaments are typically nucleated at the plasma membrane. Again, I repeat, microfilaments typically nucleated at the plasma membrane. Now, what does nucleated means? I have said previously. Nucleated generally means which have gathered around a central area. So the main central area of microfilament is your, where? Is your at the plasma membrane. So microfilaments typically nucleated at the plasma membrane. Where we have seen in microtubules, they have extend out from the centrosome of a cell, but microfilament, their main center point is at the plasma membrane. So therefore, the periphery of a cell generally contains highest concentration of microfilament. Periphery means edges. So the edges of the cell contain highest amount of microfilaments, right? Okay. Now there are a number of external factors and a group of special protein that influence the microfilament characteristics, and depending on the function of that particular external factors, they make rapid changes in the structure of the microfilaments, even if the filaments must be completely disassembled in one region of the cell and reassembled in somewhere else, right? So generally different proteins interact to form this type of association and disassociation of the microfilaments. Now, the association of actin into cytoskeleton network has been found to be of four types. So there are four types of actin protein, which not four types of actin protein. We have seen the actin protein associate with the cytoskeletal network in four ways. The first way is association of the actin molecules to form the actin filament, right? Which are the microfilament? Association of the actin molecules with actin filament is the first way that actin molecules they bind. The second way is your association with non-actin proteins into microfilaments. So the non-actin protein bind with the microfilaments, also called as actin filaments. The third way through which actin protein interact to with associate is joining of microfilaments with a network. Joining of microfilaments with network. So there is a cross linking now. The actin proteins are involved in cross linking now, right? Joining of microfilaments with network. And the fourth way is association of actin fibers with other cell components like membranes, because at the periphery we find highest concentration of the actin filaments to be associated. So this is a cross linking. The third part is the cross linking of the actin proteins to form a network of different microfilaments. So one microfilaments with another microfilaments are joined by the actin molecules, and individual microfilaments are made up of actin proteins as well. Non-actin proteins also associate with the microfilaments as well. And the fourth way is association of actin fibers. Association of actin fibers. So the actin fibers, now we say as actin fibers or F actin, with other cell components. With other cell components. Okay. So in this four way actin proteins bind. Now this role of actin as a supporting helper of various cytoplasmic structure, it gives an idea that assembly of actin filaments and their association with cell components are responsive to cellular controls. So in this four way as it interacts, so depending on this fact, these actin filaments help various cytoplasmic structures to associate and form, and different depending on different state of the cell, it helps in the movement, it helps in the maintaining the structure. Now there is a chemical it's actually a metabolite called cytocalasin. Cytocalasin, This cytoskeleton is a metabolite derived from a fungus called Helminthosporium. So this metabolite is derived from a fungus called Helminthosporium. Okay, now it is saying that this cytokalasin have a profound effect on these actin filament, just like um, we have seen in case of microfilament, that colchicine, then your vimentin, then your, sorry, not vimentin, colchicine, vincristine, vinblastine. These are the drugs which inhibit the microtubule assembly, right, and causes disassembly. Here, cytokalasin derived from the fungus Helminthosporium. This cytokalasin and its metabolites, they have an effect on the actin filaments at a very low concentration of about 1 into 10 to the power 9 m. It inhibits the further addition of actin molecules to the microfilaments. Okay, it causes fragmentation of the microfilaments as well. So it causes fragmentation of the actin filaments. It induce it also inhibit that, so that no actin filaments will assemble to form microfilaments. Okay, now there are some proteins also which your link which inhibit the cross linking between the actin filaments. So if these are the intermediate filaments and actin proteins are associating, some proteins are inhibiting this cross linking of the actin filaments, or also they are inhibiting the uh, actin filaments to associate with the membranes. Okay, so different examples are there which cause this type of inhibition, right? Some protein are also there which binds to the end of the microfilaments and inhibiting the growth of the filaments. There are also some proteins which bind to the ends of the microfilaments and inhibiting the growth of the microfilaments, and these proteins are called as capping proteins. So they bind to the uh, ends and prevents the growth of the filaments, called as capping proteins, like fragmine. These are the proteins which cause this type of action, capping proteins. Spectrin. Then comes beta actinin. Then comes valine. So these proteins actually bind to the polar ends of the microfilaments and inhibiting the growth of the filaments. I have discussed with you little bit ago that they have a plus end or positive growing end where these proteins will bind and prevent further assembly of the microfilaments. Most of the actin binding proteins plays an important role in binding of the microfilaments to the membrane. I have said that many proteins, they changes the not orientation, they changes the state of the microfilaments assembly, disassembly. Okay, so detailed studies has been done in the microvilli of mammalian intestine to find out the relationship between the membrane and microfilaments. Longitudinal section of this microvilli, which is found within the intestine, have shown the presence of long thread-like structure of microfilaments like microfilaments in the villi. Okay. Now, if I say the chemical composition of the microfilaments, I have already said that actin is the main structural protein. So, your actin is the main structural protein of the microfilaments, and the concentration of the actin in non-muscular cell is also high. It may account up to 10% of the total cell protein. Okay. So actin are the main proteins found in the microfilaments. This I have already said. But I have not said that there are three types of actins called as alpha, beta and gamma. The alpha form of actin is found in fully mature muscular tissue. The other two forms are more characteristic of non-muscle cell. So the alpha form of actin is found in fully mature muscular tissue, right? Muscle tissue. Within the muscle tissue, we will found this type of mature alpha actin, whereas the rest of the two, beta and gamma, they are more characteristics found in the non-muscle cells, right? Now in non-muscle cells, the microfilaments, being of actin composition, can bind with myosin. Myosin within the non-muscle cells, the microfilaments made up of actin bind with myosin. Myosin is a superfamily of proteins which can bind with actin. It hydrolyzes ATP, okay, and transduce a force, right? Myosin is a contractile protein actually. Okay. Now in vitro studies has been done about this association of actin with the myosin at the non-muscular cells in situ and in vitro. Within the lab, it was observed that microfilaments can be coated. Microfilaments which are made up of actin filaments, it can be coated with heavy myosin, right? And this binding of myosin will give you a arrow like pattern because they have a polarity, right? Actin proteins, they bind in a polarity way, in a unique way. So that, and the myosin are also interacting with the actin filaments in the non-muscle cells. So when in vitro these microfilaments are coated with heavy myosin denoted by HMM, it has been said that the binding result in an arrowhead like pattern in which the arrowhead is all at the point of same direction, and this pattern indicates that microfilaments have a polarity. So if I draw the diagram, it will be look like this. So I am using different colors now. Okay, so this is your, suppose this is your actin. Okay, this is your actin. Okay, and we are adding with heavy myosin. What you will get? What we will get? We will get like this structure, this way, and it is binded with myosin this way. So here we can see we have this is a type of mode of decoration of actin filament by the myosin, heavy myosin. Okay, so the heavy myosin have bind in a same pattern which shows the polarity of the actin filaments actually. Okay, so these heavy myosin have coated with the actin filaments and they bind in a same pattern. That's why it looks like an arrow head. Okay. Why I have said about the heavy myosin, why I have said about the in vitro experiments done by actin filament binded with heavy myosin. Actually, by using this myosin, we can able to locate, we can able to identify and localize the microfilament in any type of cell. Okay, understood? Because the other filaments, like we will discuss later on about the intermediate filaments intermediate filaments are not bind by the myosin, so we can able to distinguish the actin filaments with the intermediate filaments by using this heavy myosin. It will bind with the actin filaments in muscular cell or in non-muscle cells, thus we can able to identify. Okay, now if I say about the function of the microfilaments, I have discussed with you, I have said in a brief way. Okay, but if I say in a in a point wise manner, microfilaments are found in movement associated with furrow. So microfilaments are helps in the movement associated with furrow. What is furrow? Well, furrow is a shallow region of the cell surface that begins the progression of cleavage by which cell will undergo cytokinesis. So, a furrow type of thing goes to be developed. If this is the two daughter cells, they are going to be split out. So this is the furrow formation that occurs now, and it will get pinch off. So this is known as cytokinesis, the process, separation of the cytoplasm among the daughter cells. And this movement is also mediated by the microfilaments, right? It also helps in cell migration during embryonic development. During embryonic development, cell migration occurs. So it also helps in cell migration during embryonic development. Okay, so these two functions are associated with the microfilaments. There is one more function, cytoplasmic streaming in plant cells. Cytoplasmic streaming in plant cells, as we found in Nightilla in Chara plants. Okay, so this was all about the microfilaments. I hope you have understood. I will provide you the notes just after uploading this video. Just go through these notes, just go through these video lectures. I'm repeatedly telling you, most of you are not watching this video. Okay,